This is the PowerPoint for nucleic acid structure, DNA replication, and protein synthesis. But this is part 1, so it will cover nucleic acid structure and DNA replication. So the structure of DNA is the title to this part of the PowerPoint. And what do you see here? On the left side, you see a plant. On the right side, you see animals. And in the middle, you see DNA, right? So what I need you to know is that plants have DNA, animals have DNA, and did you even know that viruses that are not alive also have nucleic acid? So everything alive has DNA, and even viruses that are not alive have nucleic acid, which is DNA, nucleic acid. So DNA is often called the blueprints of life. In simple terms, DNA contains the instructions for making proteins within the cell. So in case you don't know what a blueprint is the paper that says what your house is going to look like. It literally is blue, and then it has lines that says, oh, this is the restroom, and this is going to be a closet, and this is the main room, the bedroom. So that's a blueprint. And in you, it is your instructions for your cells. Think of it this way. Can I make a cake? So I'm going to put cake. Can I make a cake if I don't have the ingredients or the materials? No, I cannot make a cake without the ingredients or the materials. So can I make more cells without the instructions? No, I cannot, because I don't know, well, what color is that skin gonna be? What color and shape is that hair going to be? So I need the instructions. DNA is important because it's essential in all in all animals, and all plants and everything that's alive. Remember we said that? So it's in all life on Earth. And remember, viruses are not alive, but they also have nucleic acid. Also, it's important for medical purposes, such as cures and diseases. We can manipulate or change the DNA now, and we can start coming up with cures like Alzheimer's or maybe even cancer in the future. And of course, for better food and crops. Now, I don't know if you know, but we end up making the plants and animals the way we want them. If we want them to have more meat, then we will mate or breed the animals that have more meat, and we'll get babies that have more meat. And from there, the same thing. We get babies that have more meat from those parents, and then we continue to get those parents so we can have those babies. And then the plant as well. The tomato is really small. The little, have you ever seen the salad tomatoes, those really tiny ones? Well, those are really small. But how do we end up having these like huge tomatoes all of a sudden? Well, we end up mating again the little tomato with the little tomato, but the ones that are the bigger ones, and they end up having a baby. And then those babies are a little bit bigger. And we end up mating those babies, right? We get the sperm, which is actually the pollen, right? And they end up having that after a long time. It doesn't happen overnight. It happens after a long time. All right, chromosomes. What are chromosomes? Do you know how Jesus is named Chuy or Jose Pepe? Well, chromosomes are the same thing. Chromosomes are another name for DNA. Only chromosomes look like little X's. You see them right here. And these chromosomes are tight, 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 tight. And so they're called chromosomes, but they're really DNA. So they are DNA that are tight, 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 tight. They are made up of DNA. It's the same thing. Now, where do you find these chromosomes? Where do you find this DNA, this nucleic acid? In the nucleus, right? In your cells. So nucleic acid is found in the nucleus. Now, the shape of the molecule, it's a very long polymer. The basic shape is like a twisted ladder or a zipper. It's called a double helix. Now, how many sides does it have? Can you see it? 1, 2, Here is one side and here is another side. So that's two sides, double, right? And helix. Think of a helicopter, how it goes round and round and round and round and round, right? The helicopter, the top of the helicopter, the propellers, they go around. Well, this is called a helix in the DNA, how it goes round and round and round. That's why it is a double helix, because it has two sides that go around and around. And there you see it again, they're twisted together. Now, the backbone of the molecule is alternating phosphates and deoxyribose sugar for DNA. Yes? So the D is for deoxyribose sugar. And remember, sugars come in this shape. What shape is that? A pentagon. Sugars are either going to be pentagons or hexagons, but this one is a pentagon, okay? So it's alternating, which means phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, right? That's what alternating means. Every other one, right? But it's over and over again, the same thing. And what does a backbone do in your body? It makes you upright, right? But it also supports you. It supports you. And remember, the most important part of the DNA, if I make it look like a ladder here, right? The most important part of the ladder are these little steps right here. Yes, and these are called bases. Have you heard that song? It's all about the base, right? So it's all about the bases. The bases have a different pattern. You see how they look kind of different? Well, the pattern tells you you're a boy, you're a girl, you're a cat, you're a human. And ATP also looks like this. So does RNA. So let's take a look at that. So here you see nucleotide. One deoxyribose together with its phosphate and base make a nucleotide. So remember, DNA is like a wall. Yes, it's big. The wall is big. But the nucleic acid, the DNA, is made up of a lot of little parts called nucleotides. Think of a wall being made up of bricks, right? So the nucleic acid, the DNA, is made up of nucleotides, and it's made up of a phosphate, sugar, base. This would be the brick in the wall. Phosphate, sugar, base. Yes? And here you have a deoxyribose, which means it does not have an oxygen on this side. It only has it here. You see it? It doesn't have it over here. But if you take off the deoxy, it's ribose. Oh, it doesn't want to cover it. All right. So let's see if we can see it now. Okay, so if we take off the deoxy, it's just ribose, and this is RNA. Please make sure you realize that deoxy is DNA, but if it's just ribose, it's RNA. So the only thing that changes is this right here, and it will have an oxygen right there, okay, in the structure. And this is going to make a difference later on when we talk about protein synthesis and the bases that are in RNA. And also, you see ATP up here because ATP is energy. Remember? And this is triphosphate. You see 1, 2, 3 phosphates. If you also add these phosphates, it's what ATP looks like. So DNA, RNA, and the ATP have that nucleotide that is like the brick that makes up the wall of a nucleic acid. Let's continue. So one strand of DNA is a polymer. Poly means many of nucleotides. One strand of DNA has many millions of nucleotides. So it's exactly what I keep saying over and over again. This is a nucleotide. That's a brick. This is another one. That's another brick. This is another one. That's another brick. This is another one. Another brick. Another one. Another brick. And together, all of this is the wall that is a nucleic acid. DNA or RNA. Depending on what again? Depending on the sugar. Remember? All right, so very important. Remember, it's all about the base, the steps of the ladder, the stuff in the middle, right? And then the phosphate sugar, phosphate sugar, phosphate sugar. That's going to continue. That's just to support the bases. So make sure you drew a nucleotide on your notes. And know that the only difference between DNA and RNA is the sugar, right? It's either deoxyribonucleic sugar or ribonucleic sugar, right? All right, these are the four bases. It's all about these bases. So cytosine is for C, thymine is going to be T, adenine is A, and guanine is G. Remember that. We're probably going to say CTAG. We're not going to say the entire word. For example, Patricia is Patty, right? So instead of saying cytosine, we're going to say C. But how many are there again? Four. And the pattern are very important, okay? Or is very important. Make sure you remember that. Now, two kinds of bases in DNA. We have pyrimidines, which are single and they're smaller, and purines, which are double and they're bigger. Okay? So very important. You know how we have the phosphate sugar, phosphate sugar, phosphate sugar? And then on the other side, we also have phosphate sugar, phosphate sugar, phosphate sugar. Because how many sides does DNA have again? It has two sides. And the backbones don't change. You have here the phosphate and the sugar and the phosphate and the sugar, and it keeps going. And on the same side over here, you have the same thing. Okay? And this is the backbone. But what's the most important part of the ladder? The stuff in the middle, right? That's what you use to go up the stairs. So the stuff in the middle. And these are going to be the bases. Which are these over here? Pyrimidines and purines. Now, if I put two little ones in here, what's going to happen? Se caen, they're going to fall. If I put two big ones, they're not big enough. I mean, they're too big, right? They're not going to fit. So we're going to need a small one and then a big one so that it can fit. Okay? And it'll fit perfect. So think of el chiquito cuida al grande, or the big one takes care of the little one. It's always together with a big and a small one. Okay? Now, pyrimidines, how do I remember that those are going to be small? Single ring. Well, I think of this. I think of perrito, perro, or puppy, dog. Purines are the pure or the big ones. Think of perro or dog. The word is small, but the animal is big, right? Pyrimidines. Perrito or puppy. The word is bigger, but the animal is small. And the pyrimidines are going to be T and C. And the purines are going to be A and G. And remember I told you that a big always goes with the small? So the big A is going to go with T or C, one of these. And the big G is going to go with one of these, T or C. Now, they're all about the base, right? So it's going to be in the middle. You see the parts in the middle? They're going to be the different. Notice how the phosphate sugar are all yellow, green, yellow, green, yellow, green. Those don't change. But the patterns in the middle change because that's what it's important. That's what's important. Now, they're connected by hydrogen bonds, okay? And notice here you have a big one and small one. So if I haven't told you yet, the A goes with the T and the G goes with the C. Here I have a G and a C. So then here it has three hydrogen bonds. 1, 2, and 3. This is curvy. So I can say that these are G and C. But remember, which one was the purine, the big one? G. And the little one? C. So that's G and C. Hydrogen bonds are weak, but there are millions and millions of them in a single molecule of DNA. So the hydrogen bond is going to be in between the bases. And they're weak bonds. A and T have 2. So here are A and T. And you already saw that the G and C have 3. So these are 2. A was the big one, the purine, and T is the little one, the pyrimidine. And they're always going to be together. How do I know that they're always going to be together? A is a straight line. T is a straight line. Or you can say your aunt is with your tío. You see how it's straight, 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 and then the T is straight, straight. And the C and G are curvy. So you see how it's curvy, round, and the G is curvy, round. And you can say that your cat is your gato in English and Spanish, or your cookie in English is your galleta in Spanish. Someone told me your chicken is your gallina. Sure. Whatever it takes for you to remember that the A goes with the T and the C goes with the G. There are only 4 bases. Now, this is a quick review. Again, chromosome. If you look at the chromosome, it looks like a piece of yarn rolled up, only it's not a ball, it's like a little X. This yarn, you unravel it, you take out a little string, and it looks like the DNA that you see next to it. Yes? So from chromosome, you go to DNA when you take out the string. But remember, chromosome is DNA. It's just tight, 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 like a little ball of yarn DNA. Okay? And when you unravel DNA, you unravel that string, you have what looks like a ladder. And in here, the A, T, T, A, C, G, G, C. What is that? What's that? The base. Remember, there are only how many? 4 of them. And A goes with T. T goes with A. G goes with C. And C goes with G. Very good. So again, deoxyribonucleic acid, you need to remember that they are nucleic acids, right? You see that right there? Nucleic acids. Now, D is for deoxyribo. It's a sugar. If I cover this up, though, then I'm going to have ribonucleic acid, and that's going to be RNA. They are both nucleic acids. The only thing that changes is the sugar. Is it a deoxyribose or a ribose sugar that it has? Now, genetic materials of cells, genes. You need to know what genes are. They're units of genetic material that codes for a specific trait. So, they're your DNA. They're your instructions in the DNA specifically. Okay, and what is a trait again? Remember, a trait is a characteristic in your body. So, for example, the color of your hair, the shape of your hair, the color of your skin, these are characteristics that you got from your parents or your genes or your DNA. And remember, a nucleotide, phosphate, sugar, base, right? This makes the nucleic acid, the DNA or the RNA. And here's another picture of the DNA nucleotide. Phosphate, sugar, base. Again, the bases. A goes with T. And G goes with C. Remember, they're straight or they're curvy. And this is for DNA. You see that D right there? For DNA. But if I were to cover that up, remember, you would only see RNA, ribose nucleic acid. And you see this ose, like glucose, that means that it's a sugar. Okay, so this pentagon is a sugar. Okay, let's talk about the history. So, a long time ago, Frederick Griffith discovers that a factor in diseased bacteria can transform harmless bacteria into deadly bacteria. So, before Frederick Griffith, there was a man named Gregor Mendel. And Gregor Mendel worked with the pea plant, el chicharo. And he would use it on plants. And he was like, these plants look like the ones before them. Something is happening, right? The parent plant and the child plant, right? We now know that this happens in us. So Frederick Griffith also discovered this scientifically in bacteria. He said, what? There are these bacteria that are diseased. Here they are. That's a bacteria. It's diseased. And then there are these other bacteria that do not cause disease. So here's the other bacteria, right? That's the DNA inside. The other bacteria, here's a flagella. All of a sudden, the bacteria that weren't diseased, they had some of these diseased information. Like what? How is it that they became like these? What happened here? Something happened. Well, they were able to get the DNA from the disease bacteria, these harmless bacteria, and they also became deadly. So Frederick Griffith discovered, hey, something is going on here, not just in plants, but in animals and in different types of cells too. And there were these people called, they're not here, I'm sorry, but Hershey's, Hershey and Chase, kind of like chocolate, right? Hershey and Chase were two different scientists also that discovered, hey, you know that it's DNA that you get from your parents? Because people used to think that it was protein. Why would people think it was protein? What is protein in your body? Your hair, your nails, your skin, your bones, your muscles. So protein is everything that makes you you, your body, your structure. So it's not totally wrong to say or to believe that you get your protein from your parents. But do your parents give you their skin? No, they give you the DNA to make that skin. The instructions to know the color or the shape, right? So Hershey and Chase discovered that it was DNA, and they discovered this by using a virus. Anyway, after these scientists, there was Rosalind Franklin, and this lady used X-rays to take pictures of the DNA. And she discovered the shape, that it was a double helix. It had two sides and it went around and around and around like a helicopter. And her partner went and gave the information to these men, Watson and Crick. And they were able to describe the DNA and they got the credit for it. What? Yes. So these men, what they did was they were just hanging things from the ceiling. You can see it in this picture. And they were like, okay, what do we do with this? What do we do with the phosphate? What do we do with the sugar and these ATGCs? And then she, which I'll show you the picture later, she saw an x-ray of this. It looks something like this. What is that? Can you tell DNA by that? I can't. But what she was able to see is that it's the two layers. Here is one side, here's another side, right? And it goes around and around. So she was seeing it like from the top, not from the side. If you see it from the side, DNA will look something like this. Around and around and around and around. You'll see a picture in a bit. If I can get to it. There it is. You see that? How it goes around and around and around? She was seeing it from the top up here. So to her, she was only seeing this side and then this other side. And it looked like a circle because they overlapped. Anyway, they were able to make this puzzle together after that, with the help also of this man, Erwin Chargaff. And in 1947, he pretty much discovered that it's like a puzzle. A goes with T, G goes with C, and they called it Chargaff's rule. And your note should have the picture of the x-ray. Now, if A goes with T, and I have 30 A's, how many T's do I have? Well, it should also equal 30, right? And 30 plus 30 is 60. So if A, T, G, and C equaled 100, I'm going to subtract 60 from that, and that leaves me 40 of G and C. But how many G and how many C? I'm going to have to divide that 40 by the 2, right? Because they're equal. So 40 divided by 2 is 20, right? And 20. So then I should have 20 plus 20 plus 30 plus 30 should equal the 100. All right, so this man discovered that, and it was able to, again, create this DNA model that you see between Watson and Crick. Okay, so again, T goes with A. And how many hydrogen bonds in between? You see the little lines? 2. And G goes with C. And how many hydrogen bonds again? 3. All right, so again, the pure ones, the big ones are the A and G. And they always go with the little one. the pyrimidines, T and C. So then A goes with T, right? A big and a small one. And G goes with C, a big and a small one. The straight ones go together, the curvy ones go together. And those are the hydrogen bonds. G and C have 3, A and T have 2. And this is good to know because it makes us different, guys. These ATs, GCs, the pattern makes us different. And we're constantly mutating or changing the DNA. So here the opposite would be what? The opposite of the A or the complementary base is T. The complementary base of T? C. Oh, I'm so sorry. A. The complementary base of C is, there you go, G. The complementary base of G is C. The complementary base of T is A. The complementary base of A is T, and so on. You keep going. Please make sure you finish this in your notes. And you need to know that these are a code or a codon because they're the instructions or the code for you, which is different than your neighbor, right? The person next to you. So every how many is a code though? Every 3 DNAs are a code. So 1, 2, 3 right there. 1, 2, 3 right there. 1, 2, 3 right there, and you keep going. Please make sure you write these lines on your notes. Again, the chromosomes, right? They are wrapped around like a yarn. They're wrapped around something called histones. To review, at the same time that Franklin, the lady, was doing her research, 2 scientists named Francis Crick and James Watson, the men, were trying to understand the structure of DNA by building models of it. They were getting nowhere. Early in 1953, Watson was shown a copy of Franklin's X-rays patterns by her partner, not by her, and he immediately realized Watson, right? How the DNA molecule was arranged. Within weeks, Watson and Crick built a model that showed the structure of DNA. It explained how DNA could carry information and how it could be copied. So they took her information and they used it. This is what she saw. She saw that it appeared that there were nitrogen bases at the center of the molecule. Let me show you where. So right here, this is the A and T, and then G and C. You see how they're partnered up? Yes, maybe I should choose a different color for the G and C. So this would be G, and this would be C, and the other ones are A and T, right? They equal each other. And then she saw also that it twisted around like a helix. So she saw this part here, and then this part's in the bottom. But it overlapped and it showed a circle to her. So like if you're on top of a stairs that go around and around and around, right? These spiral stairs, and you're at the very top, it looks like a circle to you. She was looking at it like through the top, not through the side. So she saw 2 strands. Where are the 2 strands? This is one strand. You see it looks like an X. That's one strand. I'm sorry, this right here is one, and this is another one, and it goes all the way down. And because it goes in the spiral, here's it again, and here is it again. So it's turning. This one goes over here, and this one goes over here. But how many sides are there? There's one side, which is a different color. There's one side that's turning, and here's another side that's turning. There are 2 sides. Okay. So Watson and Crick described the DNA molecule as a double helix, which are 2 strands. So in 1953, Watson and Crick solved the structure of DNA. In 1958, Rosalind Franklin dies of ovarian cancer at the age of 37. In 1962, after she had died, Watson, Crick, and Wilkins, her partner, win the Nobel Prize. Maybe she would have won also the Nobel Prize if she was still alive, but unfortunately she died, and they don't give Nobel Prizes to people who are dead, so she did not receive anything and inherited it for her family. So, why is this important? DNA replication. In order to make more cells, we have to make more DNA. Remember I asked you in the beginning, can you make a cake without the ingredients? Can you make a cake without flour, sugar, eggs? No, you need that. So can you make more cells without the instructions of what cells it's going to be? No, you need the DNA. So how do we do that? How do we make a copy of DNA before we make a copy of your skin or your hair? Well, number one, this is the DNA. And you see how here it separates? It's going to separate so you can get the information. Number two, you have one side here and another side here. And you're starting to make a new DNA. But notice that in number three, how many DNAs do you have? Two. And you have an old side and a new side. Do you have two pinks here? How about here? Do you have two pinks here? No, you have a pink and you have a purple, right? The purple was the old one, a number one. Do you see it there? It just separated and you kept it. So this is called semi-conservative model. Semi means half. So conservative means that you're going to keep. You're going to keep one side. And the other one, the other half is going to be new. So you keep the parent. Is it really a mom or dad? No. You keep this original one. And you see how it's right here? And you're going to make only one side new. So when you make DNA, are you making two new DNAs? No. You're making one new and one old, and one new and one old, or original, right? These parent ones are the original ones, the first ones. And you keep going. So from here, you came with an old one here, and then you made a new one. And then the same thing. You keep an old one and you make new ones, and they keep going. So here, I need you to know that this part of DNA on the top and on the bottom are the same. Do you see how they did not open? Because let's pretend that DNA is from this side to this side. All of this is DNA. If I just need to copy your hair, am I going to need all of this? No, I just need maybe from here to here because that's your hair. The rest of them is your eyes, your skin, right? I just want your hair. So I just want these sections. Yes? Okay. So then what am I going to do? I'm going to open at something called a replication fork. If you ever see a fork on the road, you start off and then you split up to two different sides, like a fork. Well, that's why it's called a replication fork. And replication, remember, is going to be semi-conservative. So half is going to be new and half is going to be old or original. What do you notice here with these arrows? Can you see these arrows here? One is going up and one is going down. We're going to talk about that right now. This is called anti-parallel. What is it called? Anti-parallel. Anti means opposite. And parallel means it never touches, right? So they're going opposite ways, right? Good. And here you're making DNA polymerase. Polymerase, yes? So poly means more. So you're making more DNA. Now, how does this happen? This happens with something called helicase. And I know this is an enzyme or a helper because of the word ACE. Yes? ACE is an enzyme or a helper. And if I am Jose's, I am helping Jose. Can I help Jennifer? No, Jennifer, you need to go find Jennifer ACE, which helps Jennifers. What if I have a Pedro? I am Pedro ACE. Can I help Juan? No, Juan, I help Pedro ACE. So Pedro's, you have to go find a Juan ACE. So what is helicase helping here? The helix, right? And the helix is that little turn of the DNA. And what is it doing to it? It's unwinding and separating it. It's untwisting it, right? So as it untwists it, it makes more DNA. That's called DNA polymerase because you're making more DNA. And here you see the DNA untwisting, and you see an original on the top and the bottom, and the new ones are in the middle. So let's go over this a little more. Again, DNA starts here at the 5 and ends here at the 3. What do you notice about this sugar here and this sugar here? They're backwards. Yeah, do you see the O is in the top and then it's at the bottom? And then over here you see a 3 and a 5 because they are, one's going like this and one's going like this. They're anti-parallel. Remember what that means? They never touch, but they're opposite. So if I have a 5 here, I'm going to have a 3 here. Okay? And if I have a 3 here, I have to start at the 5 here. Now, very important. It unzips, right? It unzips. So if it's unzipping this way, let's just say, if it's unzipping this way, it's opening up the DNA. This side is easy because as it's unzipping, it is copying. Let me get this one. So copy, 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 copy. You're copying the side, right? But this other one, let's say it unzips the other way. So if it unzips from here to here, it's going to have to go, okay, copy, copy, copy. And then it keeps going, right? This is going to keep going here. It's going to unzip from here to here, copy, copy, copy backwards. And it keeps going. So this side is backwards. What? Yeah, you have to copy backwards. So this is why we have different mutations sometimes or changes in the DNA because it's copying one side correctly. That's the leading strand, by the way. You're leading the way. Leading strand. The other one's going to be the lagging strand because you're dragging behind. Okay? And then they have to tie each other up here with something called Okazaki fragments. Yes, the man Okazaki, he's the one that discovered it. All right. And so in the end, it should look like that right there. Yes? And so we do have errors in the completed DNA amounts only to 1 in 10 billion nucleotides. We do have some mutations, right? 6 fingers maybe, or a different color all of a sudden, white when you have like, brown hair. Things like that happen all the time, guys. Not to everyone because we do have DNA check itself, but to some of us, okay? All right. So make sure you complete these notes and you're going to have a part 2 and turn them in together. Part 2 will be on protein synthesis.